Let's dive into the world of Archaea. And I mean that kind of literally because, in fact, if we were to go to the coastal waters off of Antarctica, we would find that 34% of the biomass in those waters is comprised of Archaea. In fact, tiny little Archaea. So the cell shapes of, or cell sizes of Archaea can be very, very tiny, all the way down to 0.1 micrometer. So imagine that. Most of you estimated that the size of your bacteria were somewhere on the order of between 1 and maybe 5 micrometers. So imagine how itsy bitsy 0.1 micrometers would be. The shape can also vary amongst the archaeal world. We can see things that can become very, very long and very, very large. So for example, you can get, in some cases, up to 100 micrometers in length. And sometimes that is just due to them forming these long aggregates of cells, but sometimes it's just due to very long cells. So we can see cocci, we can see rods, spirals, lobed, plate-shaped, and Gabby's all about that. So she's going to join us for the lecture tonight and a little bit of archaeal action. Maybe she's thinking on the order of pleomorphic cells, remembering shapeshifters. So we see all manner of shapes and all manner of arrangements as well, sometimes seeing filamentous arrangements and aggregates. Now this may start to remind you of the fungus among us lecture, and you may think, oh gosh, I thought that it was really only the fungal world that was present in fragmented sort of reproductive measures, right? Remember how in certain of the fungal spores we would see an actual fragmentation. Remember the arthrospores that would form in the formation of asexual spores, and we would see that being typified by the fungal world. But guess what? When we think about it, this makes a lot of sense, because remember how closely we could put the eukarya to the archaea, and we are not surprised to see that they share features, right? So they can reproduce also by binary fission, and that's what we've seen throughout our bacterial adventure as being the primary means of reproduction. Metabolism is very diverse, but one thing that is going to blow your mind is that they do not have PFK, right? Get it? They do not have PFK. So that means that that really heavily regulated step, the one that PEP, feedback inhibited, that doesn't exist within the archaea. So they have to have other means of breaking down glucose or alternative glycolytic pathways. Guess what? They also lack the enzyme that catalyzes the transition step, or you'll remember that particular enzyme to be pyruvate dehydrogenase, the massive enzyme complex. They don't have that one either. So they have to have other means of getting, if they run a TCA cycle, which many of them do, some of them have a partially functional or a fully functional TCA. So then they have to have another means of converting to acetyl-CoA from our 3-carbon pyruvate. We often say that the archaea live in extreme environments, so I'm hoping that at least some of you were laughing, probably Marcus, when I said that we might dive in to the Antarctic waters, because of course, in general, those are frozen. Of course, I know where we are with that in climate change. But they do live in extreme environments. So by our estimation, these are extremes. So if we're talking about anoxic environments, thermophilic environments, psychrophilic environments, very, very cold, these are the kinds of things that by our estimation are places that we wouldn't want to live. So we tend to say that archaea live in extreme environments. It's not universally true, but by broad measure, we tend to associate them with those sorts of environments. Now, the cell wall structure is also very different from what we see in bacteria, and you'll all remember that, of course, the only form of life that has peptidoglycan is bacteria, so they don't have peptidoglycan, and that's in large part because they do not have muramic acid. They have something called pseudomurine, which does form a very polymeric structure that is very rigid, and they do have cell walls. But they also lack those really weird amino acids. Remember how the D amino acids found their way into the tetrapeptide chain in peptidoglycan in bacteria? We don't see that in archaea. So like eukaryotes, archaea do not have those weird amino acids. They only have the L amino acids. It's kind of interesting, and I talked with somebody about this. I don't remember when, it might have been Chris, but we were talking about the fact that they'll stain either gram-positive or gram-negative, but that doesn't mean they have a traditional gram-positive or gram-negative cell wall structure. Of course they don't. Now, if we move just a little bit deeper within the cell wall, we know that their membrane is, of course, going to be, just as all membranes are, a semi-permeable mosaic layer, you know, fluid mosaic layer. But we also recognize that they do not have ester linkages in their bilayer. So rather than having, you know, the phospholipids that are linked via esters, they're linked via ethers. Yes, get it, ethers in the world of Archaea. So the C bond O bond C that we see representing an ether is what we're going to see within the archaeal world. We've probably already uncovered this theme at this point, that archaea share features with both bacteria and eukaryotes. And one of the areas in which that is the most true is within the genetics. And so if we were to look at the genome of an archaeal cell, we would see certain features that are similar to eukaryotes. For example, they have much more similar promoter structures to what we see within a eukaryote, but they have a single circular chromosome, usually. There are some exceptions to that. They usually don't have histones, but some of them do have histones that package their DNA. So again, there are features of both, and there are features that are different from either. Their tRNAs are completely different. Their ribosomes are variable, but then histones are sometimes present, and RNA polymerase enzymes and promoter sequences are more like those in eukaryotes. But as I said, like bacteria, they have a single circular chromosome usually. So there are features of both. Now, your book goes into great depth on what we just covered in a quick table, so feel free to read more, and I encourage you highly to read more about these beautiful organisms. I started teaching in 2001, and then very shortly after, I believe it was in 2003 or 4, there was the discovery of what became an extremely famous in the moment microbe. And you might have seen this picture the year that it was discovered. My students gave me this picture. And this is a picture of something that we called for many years strain 121. And it was named that because, in fact, it loves to go to the autoclave at 121 degrees, and it keeps right on living. So it is one of those rare microbes that is not killed by the autoclave. It's like, woohoo, hot tubbing. So you can do the calculation on what 121 degrees Celsius is in Fahrenheit. That is hot. So it is a member of the phylum Crenarchaeota. And the crenarchaeotes, most of them like it really hot. So things like strain 121, their preferred temperature is above boiling. And you can imagine that they find those temperatures in hydrothermal vents deep in the ocean where PV nRT, right? PV equals nRT. If the pressure is high enough, the temperature can exceed boiling. These often are also lovers of acid and are sulfur dependent, and some of them are lithotrophic, but some of them are organotrophic. So it depends upon which one we're talking about. The one that I want to talk about today is one that lives in Yellowstone, because we're going to go tour Yellowstone. And this one is called Sulfolobus. Sulfolobus sulfatericus is one that we find in Norris Geyser Basin in Yellowstone. And it is aerobic. It is kind of an interesting spherical shape. It's a little bit irregular, but it loves it hot, so around 70 to 80 degrees Celsius, and it also loves the acids that you get at Norris Geyser Basin. It tends to like to grow lithotrophically, so check that. What that means is it's going to eat inorganic chemicals for dinner, but just like us, it then rips and strips those electrons off, takes them to an electron transport chain, transfers them through the ETC, makes a PMF, taps into that PMF. You know, it utilizes aerobic respiration. So despite the fact that it eats things like sulfur for dinner, and in fact it really likes to eat hydrogen sulfide for dinner, and it creates sulfuric acid as a byproduct, it's oxidizing that sulfur in the same way that we oxidize donuts. So its donuts are sulfur and hydrogen sulfide gas. So if we go on our journey to Yellowstone, which many of you have done, maybe you come in the Jackson entrance and you head up from south to north as you head into the caldera. And maybe you go and you pass by Old Faithful, right? You stop for a little ice cream at Old Faithful, watch Old Faithful erupt, and you zoom on north to Madison Junction. From there, you head north yet again, and you find your way to Norris Geyser Basin. Right here, Norris Geyser Basin is where it is hot. It's hot a lot of places, but it's acidic here. So one thing that's very different from Lower Geyser Basin and Upper Geyser Basin is that Norris Geyser Basin is extremely acidic. So we see pHs dipping down in some places to 0, which is crazy low, right? You've seen some of those weird movies where people's flesh is like getting torn off by the acid. Well, a pH of 0 will do that kind of thing. So Norris is not a place where you want to fall off the boardwalk. So speaking of the boardwalk, let's head around the boardwalk and find our way across to Steamboat Geyser. Remember when that one went off and it on some unsuspecting illegally camped tourists? It erupted. That was like poetic justice. And then you kind of run around these boardwalk and you're going to see a lot of these geysers. But the other thing that you're going to see is some pretty sweet colors. So you're actually going to see these different zones. So nearest the source, which we might consider the geyser, well, we know there's a lot of hydrogen sulfide gas there. In fact, when you get out of the car at Norris Geyser Basin, you're going to be like, woohoo, smells like rotten eggs, because there's so much H2S. Well, not so much, right? If the smell went away, we'd know that it was too much and it was going to kill us. But there's a lot of H2S, but not so much that it's going to harm us. And so then if we look at the aqueous regions that surround these geysers, you start to see colors. And there's a yellow zone, a red zone, and a green zone. The yellow zone is where we see Cephalobis living, and it's where it's getting plenty of hydrogen sulfide to eat and, chowing down on hydrogen sulfide, making sulfuric acid, only further acidifying, and using its aerobic respiration in that zone. Now, there are other anaerobes living there too, and we're about to meet one of those. The red zone is where you're going to find some iron oxidizers, and then the green zone is where you're finally going to find the eukaryotes. Now, they're wimps when it comes to temperature. So notice that as the hydrogen sulfide concentration goes down and the temperature goes down, it's not until we get out to temperatures around like 50 degrees Celsius where wimpy eukaryotes like us can live. So it's only bacteria and archaea that live in these super, super hot temperatures. And speaking of bacteria, we not only see the archaea, but we also see bacteria in these zones. So this is a cool picture showing mostly red zone and a little bit of green zone. We can also see the red zone, yellow zone, and wait a minute, what's all this white stuff? Well, speaking of bacteria, this is Hydrogenobaculum. And Hydrogenobaculum is in that phylum Aquaphysae. And they are, so they're water makers, right? But Hydrogenobaculum is kind of forming this white, almost snotty looking sort of precipitate in there. Here's a picture of a source, and then radiating out from there, this is a great picture where you can actually see green, yellow, and red zones. And my very favorite picture is about to come. There's a nice yellow. Do you see right here near the source, yellow zone, red zone? There's not cool enough temperatures here to be any green zones. You wouldn't want to fall in there. But here we see it all. Yellow zone, red zone, green zone, beautiful colors. In the green zone, there's an algal cell called cyanidium, and cyanidium can tolerate pHs of 0, super acidic. It's just a wimp when it comes to temperatures. So let's talk about a neighbor of Cephalobis. So in that yellow zone, we also see some anaerobes. Thermoproteus is an anaerobe that lives there. It's one of those ones that I talked about earlier that gets really, really long. It can get up to like 100 micrometers. It's very, very thin and long. It can sometimes be a little bit branched, but these are very different than Cephalobis in that they do not live in aerobic conditions. They're strictly obligately anaerobic. So rather than using oxygen as a terminal electron acceptor, they're going to get along really well with Cephalobis because they're going to use the sulfur or sulfuric acid. The sulfuric acid that is made by Cephalobis is like, woohoo, my terminal electron acceptor, and they utilize that to grow using anaerobic respiration. So their donuts are more like our donuts. That is, they're chemoorganoheterotrophs. They're eating organic compounds. But then they're using the sulfuric acid leftovers from the sulfolobus as their terminal electron acceptor to respire anaerobically. This is a really neat picture. It was taken up at Carleton College. Well, I should say it was taken in Yellowstone, but by researchers at Carleton College. And this is so sweet because you can see this is a virus right here that's attacking the thermoproteus. So get that, think about that. There is a whole viral world that just they all attack Archaea. And you might want to like ponder, what is their route of transmission? You know, like human viruses got around, you know, they were respiratory viruses on, you know, if you sneeze, you might transmit a human virus. But these archaeoviruses in Yellowstone, you guessed it, they get around on steam. So the steam is like the sneeze of Yellowstone that gets them around. The phylum Crenarchaeota is pretty small compared to the phylum Euryarchaeota. And we're going to dissect this phylum just a little bit, taking a look at some sweet membership. We are going to begin with the methanogens that, of course, make methane gas, you guessed it. Colorless, odorless, flammable, totally flammable. We're going to look at the halobacteria, salt lovers. They like it really, really salty. And the thermoplasms, you're like, oh, they like it really hot. Well, guess what? More than anything, they like it really acidic. The extremely thermophilic sulfur metabolizers. Well, I guess it goes without saying that they like to metabolize sulfur, but they're also liking temperatures above 100 degrees Celsius. And finally, the sulfate reducers, boy, they contribute to that smell of rotten egg. Beginning with the methanogens, the largest group, really strictly anaerobic. So they are all going to utilize anaerobic respiration. They tend to chow down on, again, very strange things that we wouldn't particularly care to eat as donuts. They like to oxidize hydrogen gas. They sometimes, that's their preferred, you know, that's like, you know, that's the donut with the pink frosting and the sprinkles, right? It's the really good one. But the CO2, formate, methanol, and acetate are also things that they will see as energy sources. So primarily lithotrophic, right? Liking hydrogen gas. So they'll rip and strip their electrons off of hydrogen gas, and then CO2 will serve generally as a terminal electron acceptor in anaerobic respiration. And if you think about that, if you take highly oxidized carbon, right, we know CO2 totally oxidized, fully, thoroughly worn out by our standards. Well, if it serves as an electron acceptor in anaerobic respiration, we know it's going to go from having lots of bonds to oxygen to having lots of bonds to hydrogen. So we expect to get methane gas as the byproduct of reducing CO2. And you guessed it, that is the main definitive factor about the methanogens, is that they're pumping out a ton of methane gas. Now, I want you to ponder the fact that this is colorless, odorless, and flammable, and particularly the last part, flammable, and think about what are some applications of that. As I mentioned, that there are all manner of different genera in this group. Some of them are rods, cocci, spirals. There's one called Methanopyrus that likes it really hot, Methanospirillum, Methanosarcina. Of course, Methanosarcina is this one down here that forms the packets of 8. So they're really diverse, a lot of different morphologies that we see. But one area that they're not diverse is that they're always found in these anaerobic environments where hydrogen and CO2 are rich. Now, that's going to include things like rice paddies and marine sediments, any kind of anaerobic sediment in the mud. But it's also going to include our gut. Yes, our gut, the gut of us and other warm-blooded animals. We have a lot of methanogens. And in fact, you can think about it when you burp, you know, you probably... and/or fart, right? Remember joking about that when you were a little kid, lighting your farts on fire? Well, guess what? Colorless, odorless, and flammable? You bet. So if you think about it, it makes a ton of sense that methane gas is one of the things that is released from our gut because we have methanogens in our gut, but likewise in our fecal material. So really smart decisions are made at wastewater treatment plants when they decide to utilize the methanogens that are releasing the methane gas to power the wastewater treatment plant. Brilliant! Like, that just makes a lot of sense, doesn't it? And guess what? In Europe, in the UK, they have a poop bus. The poop bus, it totally rocks along getting powered from poop. Makes so much sense. Now, what about cattle? Well, rumen methanogens may cause a cow to belch 200 to 400 liters of methane per day. From 1 kilogram of cow manure, you can get 600 liters of methane per day. Now, that sounds promising if you're thinking, dang, we could generate a lot of energy from that, and we could. So I kind of have this like weird visual of cattle with, masks on that we could harness their belches, and that would be great. But until that time, we actually have a bit of a problem on our hands, because in fact, with this belching of a lot of methane gas and the production of a lot of methane gas from a lot of fecal material, this is bad news, because methane gas is far worse as a greenhouse gas than CO2. So this is adding to our problem of climate change greatly. This is a picture that one of my students took in 2010, and I just absolutely adore it. So more cow belching. Let's talk a little bit about these salt lovers. So halobacteria, they're really liking a lot of salt. They will actually live at concentrations of salt that come very near or around saturation. So when you look at this picture of a marine saltern down at the very bottom, and in fact this is Lake Owen, and I would love to go there someday. This isn't my picture, but if anybody ever takes a picture of this wicked rad colored kind of red Halobacterium salinarium, please send it to me. So this is a really amazing place for an organism to be able to live, where you can see that the salt is literally super saturated, like it's falling out of solution. And the halobacter actually, they require this in order to grow. So they require like 20 to 25% sodium chloride in order to live. Their cell walls will disintegrate if they don't have that much salt. These are mostly aerobic chemoheterotrophs, they generally degrade things that are organic, and they're well known for, in fact, being able to utilize pyruvate directly in their metabolism, so you can think about where that enters into catabolism. And they require complex nutrients such as proteins and amino acids. They're not motile, or if they are, they will be so via Lophotrichus flagella. Remember that tuft of flagella? And I already mentioned they require this tons of salt, just a high concentration of salt. Now the pigmentation is interesting because they're more likely to gain their energy by using organics than by being photosynthetic. So you can see why in an incredibly salty lake like this, they would want to have photoprotective pigments as well, because they're getting really plummeted with a lot of bright sunlight. All right, salt lovers. So how about looking for a minute at the heat lovers that we see in places like these coal refuse piles? I look at this picture and I think home sweet home, because Leadville was an EPA super fun site when I was growing up. We had a lot of coal refuse piles. I can't say that any of the coal piles in Leadville ever really got this hot, but you can see here it's smoking. And this smoke is being produced because remember, like the fool's gold in these areas, the iron sulfide. Well, Thiobacillus ferrooxidans, we know that it utilizes that as its donuts, and then it produces sulfuric acid. So the sulfuric acid then creates a lot of heat. If anybody's ever spilled sulfuric acid on a benchtop, don't do it, by the way, it's kind of dangerous, but it does get really hot and smokes. And so thermoplasma looks at this and it's like, woohoo, I love it. I love living in this really hot temperature. So the thermoplasms are chemoorganoheterotrophic, but they are heat lovers and they lack cell walls. So this is kind of weird, because you'd think living in that extreme of an environment that they would need to have cell walls to protect them. But in actuality, they use like the tetraethers in their membranes to protect them. So they just have uniquely structured membranes rather than having cell walls that would provide that protection. So thermoplasma is one genus in the thermoplasms, and it tends to grow in these coal refuse piles that contain the iron sulfide, which is fool's gold. And then that gets oxidized, makes a hot, acidic environment. And it likes to grow at pHs of 1 to 2. Now, one thing that's super interesting about this pleomorphic cell shape is that it's temperature dependent. So if they're at super, super hot temperatures, they're more of a rod, and super cold, well, they don't ever go down to super cold, but if they're at the cooler range of their temperature, they tend to be more spherical. The other thing you should jot down about thermoplasma is that they use histones to package their DNA. So this is a good example of an archaean that has some pretty quintessential eukaryotic cell features. Picrophilus is another thermoplasm, but the thing that differentiates Picrophilus is that it has an S layer, so it has that really super defined tiles-like glycoprotein layer outside of the cell. Additionally, it's differentiated by its extreme love of acid. I mean, this guy's addicted. It likes to grow at like pHs of 0. So this is super, super low pHs. It was originally isolated from a place kind of like Yellowstone, but it's in Japan. So it has this kind of irregularly spherical shaped structure. So the very last group that we're going to look at are the very thermophilic bugs that live generally in these hydrothermal vents. So even though there is absolutely no light in this hydrothermal vent, you can see that it is teeming with life. It is filled with life. And at the basal level of those food chains, we find things like the extremely thermophilic sulfur oxidizers or sulfur metabolizers. So these are very hyperthermophilic with an optimum that's sometimes around 100 degrees Celsius. You can see they kind of look like strain 121. They have that similar kind of morphology, only they're euryarchaeodes instead of crenarchaeodes. But most of them are going to utilize anaerobic respiration, and in doing so, we know that they're going to reduce their sulfuric acid or sulfur, and adding hydrogens or adding electrons onto that, which may take the form of hydrogen, forms that smelly hydrogen sulfide gas. So pyrococcus quite literally means fireberry, and that's really, really descriptive of this group. The very last archaean that we're going to end our coverage with is one that is extremely, extremely famous. This is Archaea globus, and Archaea globus got featured on the cover of Nature a while back, and the reason that it did is because it is considered like the oil refinery's biggest nuisance, so it kind of makes me joyful. But what Archaea globus does is it lives in pipes, and so it just like our extremely thermophilic sulfur metabolizers, these sulfate reducing bacteria also produce hydrogen sulfide. So you remember this from lab because when you produced hydrogen sulfide in your tubes, you saw that it turned it black. Well, that's because there was iron in the tubes, in the SIM tubes that we used. And so the iron reacts with the hydrogen sulfide and it makes iron sulfide, and iron sulfide is that black precipitate. But think about it, if this happens in pipes, that equals corrosion. So when these hydrogen sulfide producing bacteria react with the pipes, it causes the pipelines to corrode. And in oil refineries, they're at hot temperatures, so Archaea globus causes the pipes in an oil refinery to corrode, and it just sours the oil refinery. So it's kind of listed as it's a little bit of my rock star protester, right, to oil refineries, but it's a beautiful organism, and not to be maliciously maligned, because it has a lot of really great features as well, and applications of being able to chelate out metals, right? Obviously, if it reacts with iron, it can chelate out metals from solutions, so it has a lot of industrial applications in that way as well. So, story told about the Archaea. And remember that we will have a guest lecture on Friday, but because we haven't covered everything that we need to cover for you to finish your exam, I will also provide you with a farewell vodcast that covers those last few things. So have a wonderful Wednesday.